Good day, dear colleagues. My name is Vladimir Nazarov, and let me present to your attention a lecture dedicated to the determination of mutations of the BRCA1, BRCA2 genes associated with breast cancer. In the Russian Federation, breast tumor is the most common cancer type among women. In most cases, histologically, these lesions are invasive carcinoma of no special type and invasive lobular carcinoma. From the point of view of inheritance, all forms of breast cancer are usually divided into sporadic, familial, and hereditary cases. Sporadic breast cancer is characterized by the absence of a family history of this malignancy in the patient and the absence of germline mutations in the genes associated with this disease. Familial breast cancer is characterized by a family history of malignant tumors, but without confirmed germline mutations in genes associated with breast cancer. In 10% of cases of breast tumors in patients, mutations in the genes associated with the development of this type of tumor can be found. These cases are called hereditary breast cancer. Hereditary breast cancer is classified into two types according to mutated genes. One group consists of cases in which mutations cause a specific syndrome, and in addition to the main symptoms, there is an increased risk for developing breast cancer. The second group is represented by cases with mutations in homologous recombination genes, change in which specifically increase the risk of developing breast cancer and a number of other malignant tumors. This lecture will focus on the BRCA1, BRCA2 genes belonging to this group. Before we continue discussing the clinical implications of changes in these genes, let me remind you how the human homologous recombination system works. Homologous DNA recombination is a mechanism for error-free repair of the DNA sequence after double-strand breaks. This mechanism involves a wide range of proteins coded by ATM, BART1, BRCA1, BRCA2, TEC2, PALP2, RET51C, RET51D genes. DNA double-strand breaks occur in the cells of our body constantly due to the influence of environmental factors. However, normally these breaks do not lead to the cell destruction. DNA breaks are detected by the RTR-ATM proteins, which in turn initiate assembly of the complex responsible for homologous recombination. The BRCA1, TEC2, NBN, and RAD50 gene proteins are primarily involved in double break repair. This complex removes one strand of DNA, leaving a free tail of nucleotides. Proteins BRCA2, PALP2 are attached to the resulting structure, which attracts the homologous chromosome to the break site and, copying information from it, accurately restores the damaged DNA sequence at the break site. Pathogenic variants and mutations in genes involved in homologous recombination lead to pathogenic changes in the structure of proteins and dysfunction of the components of this system. In presence of mutated genes of homologous recombination system, an additional system of non-homologous recombination is activated in the cells of the body. This system does not use a homologous DNA strand to repair breaks, which results in random addition of nucleotides to the resulting defect. This DNA scar can also occur in the coding region of the genes and lead to the initiation of malignant cell transformation. After we have considered the functions of homologous recombination, let us dwell more specifically on the clinical significance of mutations in the BRCA1 and BRCA2 genes. Mutations in these genes occur in 60 to 80% of all cases of hereditary breast cancer. They significantly increase the risk of developing this cancer. The risk of developing breast cancer before 80 years in carrier of mutations in BRCA1 gene reached 72%. In carrier of mutation in BRCA2 gene, 62%. Mostly aberrations are represented by small deletions and insertions, and although they can occur in all parts of these genes, in Russia there is a high prevalence of only a small number of mutations presented on the slide. There are a number of official recommendations describing the indications for testing mutations in the BRCA1, BRCA2 genes. For example, the recommendations of Russian Society of Oncologists, RUSCO, and as well as NCCN and ESMA associations. Indications for testing differ depending on the presence or absence of malignant neoplasms in patients. For diagnosed breast cancer, testing is indicated for all patients under 45 years of age. In older patients, the purpose of the study depends on the family history of cancer. In male patients, the study of mutations in the BRCA1 and BRCA2 genes is carried out in all cases. In the absence of diagnosed breast cancer, the study is indicated for first-line relatives of patients with a detected mutation. Also, indications for tests are aggressive and metastatic forms of breast cancer. In these cases, the detection of mutation is an indication for the prescription of targeted therapy. It is important to note that the current indications for testing mutations in the BRCA1 and BRCA2 genes are not ideal. Thus, recent studies emphasize that about 40% of women with BRCA2, BRCA1 mutations do not have a family history and do not meet the NCCN criteria for indications for molecular genetic testing. This fact makes oncologists think about significant extension of indications for genotyping of these genes. What is the clinical significance of detecting mutations in the BRCA1, BRCA2 genes? Firstly, the detection of mutation in these genes confirms the hereditary nature of breast cancer, which changes not only approaches to the surgical and therapeutic treatment of patients, but also the prevention of the development of tumors in other localization in the patient and his relatives. Unfortunately, BRCA-positive cases of breast cancer are characterized by a more aggressive course with an early age of onset. However, in patients with identified mutations, it is possible to use targeted therapy, PARP inhibitors, olaparib, talazoparib. To date, in Russia, all patients with confirmed breast cancer can be tested for the presence of mutations in the BRCA1, BRCA2 genes at the expense of the compulsory medical insurance fund. DNA technology has developed a kit that allows you to identify the most common mutations in the Russian Federation in the BRCA1, BRCA2 genes. A feature of this kit is the ability to simultaneously evaluate 8 major mutations in the BRCA1, BRCA2 genes with automatic analysis of the results. It is important to note that this kit is not used to diagnose breast cancer, but is only used to detect mutations in the BRCA1, BRCA2 genes. The material for the test is whole venous blood of the patient in the test tube with EDTA. The slide shows a brief algorithm for laboratory testing of sample using a kit for determining mutations of the BRCA1, BRCA2 genes associated with the risk of developing breast cancer. Isolation and purification of DNA is carried out from the venous blood using Proba-GS genetics kit or Proba-Rapid. After DNA purification, mutations are studied using the PCR-based kit on the DT thermal cycle. The software automatically evaluates the results and prepares a detailed report. At the end of this lecture, I would like to give a clinical example that confirms the significance of detection of mutations in BRCA1, BRCA2 genes. A 34-year-old woman whose mother and grandmother had cases of early breast cancer refers to an oncologist for further examination and treatment after histological confirmation of triple-negative breast cancer. Genotyping of the BRCA1 and BRCA2 genes was prescribed. Pathogenic polymorphism in BRCA1 genes was revealed, which made it possible to choose the right therapeutic, surgical, and preventive tactic for the patient. Colleagues, thank you very much for your attention.